Regulating prokaryotic gene expression. How does a cell know what genes to express or what proteins to make? Are all proteins made at the same time? Prokaryotes and eukaryotes have different ways to regulate gene expression, or in other words, turn genes on or off depending on the cell's needs. Prokaryotes regulate the expression of genes by controlling transcription. Eukaryotes, on the other hand, can control gene expression at many points. Controlling gene expression. Individual cells respond to environmental change by regulating their gene expression. Prokaryotes and eukaryotes have evolved different mechanisms for regulating their gene expression. Prokaryotes regulate gene expression in only one part of the transcription process. Eukaryotes have multiple places to regulate gene expression, both in transcription and translation. Prokaryote regulation will be discussed in this topic. Eukaryote regulation will follow in the next topic. Regulation of gene expression in prokaryotes. Individual prokaryotes respond to environmental change, such as varying food sources, by regulating their gene expression. The control takes place during transcription. For example, when lactose is present, bacteria can freely transcribe the genes coding for the enzymes in charge of breaking down this sugary source of food. However, when lactose is absent, the transcription of these genes is physically blocked. The enzymes that process lactose do not get synthesized. This mechanism saves the bacterial cell both energy and materials. Operons. In prokaryotes, genes are clustered into operons within the chromosome. Operons consist of three parts: promoter, which is where the RNA polymerase bonds, the region where that happens; the operator, which either blocks or supports RNA polymerase; and the genes, which code for the specific protein required by the cell. So you can see this control region consisting of the promoter and the operator, and then the structural genes here. And both of these together make up the operon. The regulatory gene produces proteins that instruct the operator to either block or support the bonding of RNA polymerase to the promoter. Regulatory genes may be found outside the operon, as shown here, or they may be located within the operon. Regulatory proteins. Regulatory genes produce two types of regulatory proteins. Repressor protein, negative regulation, where transcription occurs when the protein is inactive by blocking RNA polymerase from bonding to the promoter region. Or activator protein, which is positive regulation, and transcription occurs when the protein is active by allowing RNA polymerase to bond to the promoter region. Regulatory proteins are allosteric. They only become active or inactive when they bind to the operator. Repressible operons. A repressible operon is one that is usually on. When a repressor binds to an operator, transcription is shut off. The TRP operon for tryptophan synthesis is a repressible operon. The TRP operon codes for a number of genes responsible for the production of the amino acid tryptophan. When tryptophan is present in adequate quantities, the cell can stop making enzymes for its synthesis. This conserves resources so that the cell is not expending energy to make tryptophan where there's a significant amount already present. Tryptophan acts as the co-repressor in this operon. It binds to the repressor, causing the repressor to bind to the operator to prevent transcription. This is an example of a negative feedback loop. The product, tryptophan itself, inhibits its own synthesis. Here's a visual of the TRP repressor operation. When tryptophan is present in adequate quantities, the cell can stop making enzymes for its synthesis. So when tryptophan is present, the TRP repressor binds the operator. And you see how tryptophan binds the repressor binds the operator. And when that happens, the RNA synthesis by the RNA polymerase is blocked. In the absence of tryptophan can't bind the repressor, and therefore the repressor can't bind the operator, and RNA synthesis proceeds. Repressor protein assistance. A co-repressor is a small molecule that cooperates with the repressor to help switch an operon off. We just showed an example of that, right? The tryptophan molecule itself acted as a co-repressor, binding to the repressor to then allow it to bind to the operator and block the RNA polymerase. An inducer is a molecule that inhibits a repressor, so stops that repressor from binding to the operator, and in turn helps to switch an operon on. So let's look at an example of that now. Inducible operons. An inducible operon is one that is usually off. A molecule called an inducer inactivates the repressor and turns on transcription. An example of an inducible operon is the Lac Operon, which contains genes coding for enzymes that break down lactose into glucose so the bacteria can use it for energy. If no lactose is present, then no enzymes need to be made. The bacteria saves energy this way. In this operon, the lactose molecule is the inducer. The 3 genes for lactose enzymes form an operon. They are adjacent on the chromosome, share a promoter, and are transcribed together. The Lac Operon is very efficient, but activity can be reduced when it is not needed. An example of transcriptional regulation. The repressor prevents transcription until a beta-galactoside predominates. A repressor protein is normally bound to the operator, blocking transcription. In the presence of lactose, the repressor detaches and allows RNA polymerase to initiate transcription. In the presence of lactose, RNA polymerase can initiate transcription. So you can see that ordinarily the repressor binds the operator and the transcription is blocked. But in the presence of lactose, which can bind the repressor, causing it to fall off the operator, the polymerase can move, allowing transcription.